Hello, fellow botanists. Welcome to our final lecture about plants at the organismal level, and also our final lecture on plants for the entire section of this course. So it's a little bit of a bittersweet lecture, but I hope to end with a really interesting set of observations. Final reminder of where we've been and where we're headed in this lecture. We have talked about the structure of plants, we have talked about the development of plants, and we're talking about the physiology of plants. We're thinking about how plants face the problems of regulating an internal environment. And this is part of our bigger picture of thinking about how plants solve the problems of being multicellular organisms. And as I've tried to emphasize throughout these lectures, this is all about behavior. Plants really are behaving at many different levels. They're behaving at developmental levels and they're behaving at physiological levels. In our last lecture, we talked about how plants were going to circulate or at least move resources through their body, such as nutrients, water, carbohydrates, etc. In this lecture, we're going to think about how plants exchange gases with the environment. And as with all multicellular organisms, there are two primary gases that plants need to exchange with the environment, one being carbon dioxide and the other being oxygen. However, as you probably know, plants are exchanging these gases for a somewhat different set of reasons than we would as humans. While we're trying to get rid of excess CO2 that's building up through the process of cellular respiration and bring oxygen in, plants are trying to bring in carbon dioxide for the purpose of photosynthesis and get rid of at least some of the oxygen so that they don't get an excess buildup of oxygen inside their cells. But remember that plants still do need some oxygen as well. So of all the lectures, I think that this last lecture on the exchange of gases at some level might be the most familiar to you, because at some point in the past you've probably talked about photosynthesis in an earlier class. So I'm going to do a little bit of a review of photosynthesis as I start off this lecture over here. We're going to talk about the basics, sort of a reminder, and then I'd like to show how this problem of photosynthesis generates all sorts of trade-offs. And so it's really going to be these trade-offs that the plants are having to deal with at a physiological level and as they regulate their behavior in different environments. To modulate these trade-offs, plants can respond in terms of using their stomata. And if you remember, stomata are these structures that open those little openings that can change size based on the guard cells. Stomata can regulate their opening size, but also we'll think about some developmental regulations. And so stomata is actually how plants breathe. I mean, this is a reminder that plants really are breathing organisms just like animals, and in fact just like fungus for that matter too. And finally, we'll think about how plants regulate not just their stomata, but we'll think about how plants photosynthesize and different pathways available for plants to photosynthesize. And finally, I'd like to end this lecture by thinking not just about plant behavior, but how plant behavior is a response to our behavior, and then our behavior in turn is a response to plant behavior. So we'll think about this interaction between plants and humans, hopefully as an interesting way to end our series of lectures on plants at the organismal level and how plants behave in their environment. Begin our review of photosynthesis with the equation, the chemical equation for photosynthesis. And I know that you've seen this in several other classes. So as a quick reminder, we're going to start with light energy. And these are shown over here in the diagram for your textbook. So you have light energy shining on this wonderful tree. Then this light energy is going to be used by the plant to combine carbon dioxide and water. So carbon dioxide is coming from the atmosphere. Water is being pulled up from the soil through the roots. It's going to combine the carbon dioxide and the water to form carbohydrates of various kinds, such as glucose. And we can see the glucose combined also to form molecules like sucrose being exported throughout the plant body. And finally, oxygen is going to be produced as a byproduct. So here's oxygen being released from the leaf back into the environment. Also, you may remember from previous lectures, there are two major sets of reactions that we have in photosynthesis. So our first set of reactions are the light reactions, and our second set of reactions occur in the Calvin cycle. So this diagram from your textbook shows a little model of a chloroplast, and the first set of reactions are light reactions. So these are the reactions that capture the light energy using chlorophyll or some sort of other photosynthetic pigment. Water is also used in these reactions. There are a couple of photosystems. We won't go into the details there, but these photosystems are going to be responsible for splitting water using the energy from an electron in water to make to transform that energy into different energy molecules such as ADP and NADPH, and then oxygen is going to be produced as a byproduct. Then in the Calvin cycle, we're taking the energy that we produce from the light reactions, and we're using that energy to grab a carbon dioxide molecule. We're taking several carbon dioxide molecules per cycle, in fact, and we're producing a 3-carbon molecule over here that then can eventually be transferred into glucose and eventually converted into sucrose or some other carbohydrate for export. We can connect what goes on in our equation for photosynthesis with what happens in our light reactions by looking at the reactants and the products for each of our particular sets of photosynthetic reactions. So our light energy, of course, you see that coming in here, is part of our light reaction, as is water. So here's water in our equation. Then we're using carbon dioxide in our Calvin cycle. We're grabbing it with Rubisco. We'll talk a little bit more about Rubisco, but we're grabbing carbon dioxide, and we're using carbon dioxide to produce some sort of carbohydrate, in this case over here, a sugar. Finally, oxygen is given off as a byproduct from our light reactions. So what I'd like you to be able to do, and I'll move out of the way so you can see this over here, I'd like you to know the reactants and the products for each set of reactions, because these are going to be important as we start talking about our different photosynthetic pathways. To continue our review of photosynthesis, let's next turn to leaf structure. You have a great little diagram here from your textbook, and it's kind of like a little ride on the magic school bus. So we start off big, and then the magic school bus can do things like shrinking down. So we're going to start off on looking at the leaf at a big level, and we're sort of going to shrink down to see what happens inside of it. And along the way, hopefully you'll start to recognize some of the morphology of the leaf that we've been talking about in lab and other parts of this course. All right, so here is a leaf. And we're going to look at a cross-section of the leaf. So in our cross-section of the leaf, notice that the epidermis, we have an upper epidermis and a lower epidermis here. And so these epidermal tissues, of course, are part of our dermal tissue systems, protecting the plant from the external environment. In our plant leaf, we also have veins. So you can see the veins externally, but when we do our cross-section of the leaf, you can see that there are a variety of veins. So here would be a little vein coming through the middle part of the leaf. And remember that veins have vascular tissue, including xylem and phloem. Then, tissue that is not part of the epidermis or the vascular tissue is part of the ground tissue system, and we call this the mesophyll in leaves. And remember that there are often 2 different layers of mesophyll, sort of an upper layer of the mesophyll where the cells are packed very tightly together, and so that's so they can absorb lots of light energy from above, and some lower layer where mesophyll cells are dispersed a little bit more, and so that allows water vapor and gases such as carbon dioxide and water to move amongst these cells over here. Now, of course, if we have gases, and this whole point of the lecture is to talk about gas exchange, we need to get gases into and out of our leaf, and that happens, as you hopefully remember, through these stomata, right? These little openings that are regulated by guard cells. So in general, carbon dioxide is going in and oxygen is coming out. And the stomata are on the underside of the leaf, and this is very important because the underside of the leaf, while there's not a big difference here between the top and the bottom in terms of absolute space, the underside of the leaf does tend to be appreciably cooler than the upper side of the leaf. And therefore, if the underside of the leaf is appreciably cooler, we don't get quite as much water loss through evaporation. And we're going to see in a moment that that's sort of one of these central trade-offs that we're dealing with in photosynthesis. Finally, if we zoom inside one of our mesophyll cells over here, so we've zoomed inside a mesophyll cell here, notice that there are lots of chloroplasts. And this is a sort of reminder of the structure of the chloroplast. We have an outer membrane, we have an inner membrane, we have a thylakoid membrane over here. And these are our 2 sets of reactions, our light reaction and our Calvin cycle that occur inside of our chloroplasts, either inside the thylakoid membranes, across the thylakoid membranes, or inside the stroma which is the space between the thylakoid membrane and the inner membrane. Understanding of photosynthesis gets us to where we really want to go with our review of photosynthesis, and that's to understand trade-offs. So I put once more our chemical equation for photosynthesis up here, minus light because it didn't quite fit. But let's think about what the plant has to do to get both of these reactants, to get the carbon dioxide and the water that it needs for photosynthesis. And these, of course, are being regulated by the stomata. And so it turns out that plants are going to have trouble getting both of these at the same time. So when our stomata are closed, right, plants do that to hold on to water. So that tends to be when plants encounter hotter and drier conditions. So they're hanging on to water, that's the benefit, but the cost is that when your stomata are closed, you can't get carbon dioxide in, and you risk building up too much oxygen inside the mesophyll cells of the leaf. On the other hand, you can open your stomata to bring in CO2 and let O2 out. That's the benefit, but the cost is that you also lose more water. So, as with everything in life, you can't have your cake and eat it too, or as plants would say, you can't have your carbon dioxide and water as well. So, how are plants going to regulate this trade-off? And there are a couple of solutions that I'd like to talk about. The first is that plants can regulate these stomata in a variety of ways. So, not just opening and closing them once they're formed, but also through developmental regulation. Remember, plants are always producing new leaves, they're always developing in response to their environment, and so plants can adjust the number of stomata that they put on their leaves based on the location of the leaf in the environment, based on the environmental conditions that a plant is encountering at any particular point in time. And then, sort of at a bigger level, sort of a larger macroevolutionary level, we can have various photosynthetic pathways, different photosynthetic pathways. They're going to help plants balance this trade-off between carbon dioxide and water loss. So I'll end with our little fun fact over here on this particular slide. How significant is this trade-off over here? How significant is water loss through the stomata? And plants lose about 90% of their water loss through the stomata, even though these stomata occupy only about 1 to 2% of the surface area on the epidermis of leaves. So, yeah, this water loss is a really significant problem for plants. The most basic response that plants have to this trade-off between carbon dioxide and water is the physiological response of changing the diameter of the stomatal openings with guard cells. So I'll just sort of very quickly mention how guard cells work. Here we see the epidermal cells of a leaf, and guard cells are also epidermal cells, but they perform a somewhat different function than other epidermal cells. Guard cells can exchange ions with surrounding epidermal cells. So if a guard cell starts to take in lots of ions, such as potassium ions, as I see in this picture here, then what that does is that causes a imbalance in our solutes. We have more potassium inside than we have outside. And so what happens is, if you remember your basic osmosis from Biology 111, we have a diffusion of water from outside of the guard cell inside the guard cell, and in particular inside the vacuoles. So here we have a vacuole and a vacuole, and you can see lots of water diffusing into these vacuoles. Now, as water diffuses into these vacuoles, what happens is these vacuoles are going to push against the cytoskeleton of the guard cells. And as they do that, it's actually going to warp the cytoskeleton of the guard cells, sort of pushing it open a little bit, pushing it apart, and that's going to create our stomatal opening. So water into the guard cells means that our guard cells open. And that makes a lot of sense from a physiological perspective, because if plants have plenty of water, then they're going to want to open their stomata to exchange gases with the environment. Plants are less concerned with the loss of water. On the other hand, if plants are experiencing a lower water, then what they can do is they move potassium ions out of the guard cells. And again, these can think about our regulatory processes. These can be regulated by plant hormones, as we talked about in our previous lecture, plant hormones such as ABA. So we can move our potassium ions out, as we move our potassium ions out, then water is going to osmose out of the cells as well, again following the potassium ions. And so our guard cells then go from sort of being really stretched out, sort of like an overblown little balloon, to just sort of being collapsed and limp. And this collapsing is actually what closes the opening and keeps water inside the plant leaf. Guard cells, the regulation is done by ions, particularly potassium ions. In response to modulating this trade-off between carbon dioxide and water is a developmental response, again involving stomata. The number of stomata can actually be different from one leaf to another on a plant based on the environment that it's encountering when that particular leaf was developed, or even maybe based on the location of the leaf on the plant. And certainly if we look in a population of plants, plants that are in different environments can have different arrangements of stomata on the undersurfaces of their leaves. And so these can be regulated developmentally. So I've included a little picture over here just to show you what this might look like on the undersurface of a leaf, because I've had a number of students in my lab over the years working on looking at the undersurfaces of leaves, the stomatal density, and thinking about how stomatal density is impacted by environmental parameters. So in this particular leaf over here, we see this is a monocot leaf, a grass leaf, and so we can see these little stomata sort of lined up in rows. And the really neat thing about grass plants is that the stomata sort of line up in rows right along these veins that are running through the leaf as well. So notice in this particular picture how closely placed the stomata are along this vein. Now, even if we just sort of move over a little bit and go to a different vein, notice that our placement of stomata is somewhat different, right? So for a similar length over here, we have 4 stomata versus 3. So this is just sort of a very small, really micro example of how stomatal density can shift over the surface of a leaf, but of course it can shift from leaf to leaf as well, and from plant to plant. In shade conditions, where leaves tend to be a little bit cooler, we often tend to see more stomata, because what's going on is these leaves are cooler. They don't have to worry quite as much about water loss, so they can bring in more carbon dioxide. On the other hand, when we have leaves in the sun, the stomatal density of these leaves tends to plummet. Also, stomatal density can be shifted in response to carbon dioxide concentrations. So as carbon dioxide concentrations go up in the atmosphere, plants can take in more carbon dioxide for a given amount of time that the stomata is open, and so we tend to have fewer stomata in higher CO2 concentrations, more stomata in lower CO2 concentrations. And finally, interestingly, I have to put in just a little bit of a plug for some of the research that my own students have done over the years, and they've looked at the stomatal densities based on the photoperiod, in other words, based on the day length, which is an indicator of where the plant is in the growing season. And so plants can also change their stomatal density based on when they think they're in the spring, or the summer, or the fall, or so on. So there can be a lot of developmental responses. A couple of examples here of how people have looked at changes in stomatal density can be seen both over herbarium specimens in the past 100 years and even in the fossil record. So in the past 100 years, we see changes in average stomatal densities because our CO2 concentrations are changing in the atmosphere. Specifically, they're going up. And in the fossil record, CO2 concentrations can fluctuate over extended periods of time. And so as we get fossils of plant leaves, we can actually see in some of these fossils of plant leaves stomata. And it turns out the stomatal densities tend to be very highly correlated with CO2 concentration in the atmosphere. In fact, so highly correlated that plant stomatal densities can actually serve as a proxy of the CO2 concentrations at particular points in our geologic past. Well, physiological adjustments of stomata diameters and developmental changes in the number of stomata are great ways for plants to balance this trade-off between CO2 and water. Sometimes we need something that's a bit more substantial than what we can do through these mechanisms. And therefore, plants have evolved different photosynthetic pathways. You may have heard of some of these different pathways. I'll do a quick summary of these different pathways, but again, I'll focus a little less, hopefully, on the biochemistry of the pathways and a little bit more on why the pathways are evolving as a response to managing this trade-off between water and carbon dioxide. In particular, for each pathway, what I'd like to do is consider three big questions over here, three features of the pathway. The first one is, what enzyme first binds carbon dioxide? So carbon dioxide can be bound at several different points in photosynthetic pathways, but what enzyme first binds it, first grabs it from the atmosphere? Second, in what cells does the Calvin cycle occur? So we're used to thinking about the Calvin cycle just occurring in mesophyll cells along with the light reaction, but that's not always the case. So we'll think about how moving the Calvin cycle to different sets of cells might be a good evolutionary response in certain situations. And finally, we'll think about when this binding of CO2 that occurs in the Calvin cycle, when this binding actually occurs during a 24-hour period. Is it in the day? And we're sort of used to thinking about the fact that, of course, it's in the day, but maybe it doesn't have to be in the day. So we'll think about how plants might bind CO2 at different times, such as the night. There are three major photosynthetic pathways: the C3 pathway, the C4 pathway, and the CAM pathway. I'm going to start by talking about the C3 pathway. So you may not have actually had photosynthesis, as you've talked about it in earlier classes, named, but the version of the photosynthetic pathway that you've probably been taught has been the C3 pathway. What I'll do is quickly summarize the C3 pathway. Again, I know we've just talked about it, but I'll summarize it in terms of this diagram, and I'm going to use variations on this diagram to compare our different pathways. So remember that our any process of photosynthesis involves two sets of reactions. It involves a set of light reactions, and so in our light reactions we're going to use light energy to capture water and split it to give off oxygen. And the purpose of the light reactions is to produce energy molecules such as ATP and NADPH. Our second set of reactions are the Calvin cycle, and their purpose is to take carbon dioxide, combine carbon dioxide molecules, some hydrogen, some oxygen, and then produce carbohydrates that we have that we can eventually store and move as sugars. These sets of reactions, the light reactions and our Calvin cycle, are in the C3 pathway are going to occur in mesophyll cells. So if you remember, mesophyll cells are these cells right here. They're between the epidermal layers of a leaf, and they can be somewhat spread out in terms of the vein. So they can be far away from a vein, they can also be right next to a vein. Doesn't really matter. Our first big question over here in our C3 pathway is, what enzyme first binds carbon dioxide? And so as carbon dioxide is coming into the Calvin cycle, the enzyme that grabs it is rubisco. So it's called rubisco because what it does, it takes a molecule of ribulose bisphosphate over here, and then it combines it with carbon dioxide. So we call that enzyme rubisco. Right? So it's a very important enzyme, one of the most abundant enzymes on Earth. Okay, so that starts us then through our Calvin cycle. One other note is that as soon as our rubisco binds with our carbon dioxide, we get a 6-carbon molecule. So rubisco is a 5-carbon molecule, carbon dioxide is a 1-carbon molecule. You put them together, you get 6 carbons, but this 6-carbon molecule is really unstable. And so very quickly it reverts into 2 3-carbon molecules. Why is that little note important? Because that's where the name C3 comes from, right? So it's the first stable carbon molecule in the Calvin cycle. Our second feature of the C3 pathway is we'll think about the cells where the Calvin cycle occurs. And as we just mentioned, the cell type where all these reactions occurring are the mesophyll cells. So these are reactions both where the Calvin cycle occurs and the reactions where the cell where the light reactions occur. And this is great, of course, because when our light reactions are in proximity to our Calvin cycle, we can move our energy molecules like ATP and NADPH back and forth very easily. And then finally, let's think about when during the day these reactions are taking place. When during a 24-hour period these reactions are taking place. And the answer is they're taking place during the day. So in our little mesophyll cell over here, I've indicated that the mesophyll cell can be a cell at night, the mesophyll cell can be a cell during the day, but these reactions are taking place during the day, not during the night. Our second photosynthetic pathway is the C4 pathway. So I've drawn a diagram of the C4 pathway over here, but I've put it next to our diagram of the C3 pathway to try to illustrate some of the key differences. And your textbook has a version of this figure too, but the problem is your textbook doesn't put together all three pathways, the C3, the C4, and CAM. So I wanted a diagram that would allow me to compare all of these together. So I've opted for my own here instead of what the textbook is providing. Let's start with what the C4 pathway is doing, and right off the bat it's doing something very different than what we see in our C3 pathway. So if you remember, in our C3 pathway, carbon dioxide entered a mesophyll cell and was bound by Rubisco, this enzyme that combines ribulose bisphosphate with carbon dioxide. In our C4 pathway, carbon dioxide is again coming into the mesophyll cell, but it's not being bound by Rubisco. Instead, it's being bound by PEP carboxylase. And so PEP carboxylase is an enzyme, it's ACE over here, that takes our PEP molecule, which is a 3-carbon molecule, and combines it with carbon dioxide to give us a 4-carbon molecule. And so this 4-carbon molecule is the source of the name C4 pathway. This takes place in the mesophyll cell, along with the light reactions. However, the second thing that our C4 pathway does that's going to be a little bit different is it's going to put the Calvin cycle not in the mesophyll cell, but in a new cell, a bundle sheath cell. This is the cell where the Calvin cycle takes place. Now, bundle sheath cells tend to be oriented around vascular tissue in plants that have the C4 pathway. So if you think about a core of xylem and phloem, bundle sheath cells are the cells that sort of go right around the vein, right around this core of xylem and phloem for the veins of each leaf. Our carbon molecule in the form of this C4 organic acid, 4-carbon organic acid, is going to be transported from the mesophyll cell into the bundle sheath cell. And it's in the bundle sheath cell that one of these carbons is going to be released so that it can be picked up by the Calvin cycle, so that it can be bound by Rubisco and combined with ribulose bisphosphate to launch us through this process again. So it's not as if Rubisco disappears in our C4 pathway, it's just that it occurs in a different cell. Right? And then eventually our 3-carbon molecule, returns, is converted back into PEP over here so that it can start the cycle again. Our Calvin cycle occurs in the mesophyll cell. And then finally, this is not something that's different from a C3 pathway, but all these reactions again are occurring during the day. Right? Both the binding of the carbon dioxide in the mesophyll cell, the movement of carbon into the bundle sheath cell, and our light reactions all taking place during the day. Remember that the key idea that we're trying to explore here is not just biochemical differences. We're trying to figure out how plants regulate this trade-off, and in fact maybe mitigate this trade-off between carbon dioxide gain and water loss. So what is this doing for the plant? Move out of the way so we can take a look at the advantages of this particular pathway, and there are a couple that we see that leads to sort of the big one down here. So first of all, PEP carboxylase has a much higher affinity for carbon dioxide than does Rubisco, interestingly enough. What that means is if we have a lower CO2 environment inside the cell, PEP carboxylase actually does a better job of grabbing those last remaining carbon dioxide molecules from the atmosphere than Rubisco does. In fact, if we start to get really low concentrations of carbon dioxide in a mesophyll cell and higher concentrations of oxygen in a C3 pathway over here, Rubisco actually starts grabbing oxygen instead of carbon dioxide. So this can actually lead to a form of oxygen poisoning that C3 plants can experience if they keep their stomata shut down and if they don't have enough carbon dioxide then around the mesophyll cells of their leaf. So PEP carboxylase can grab CO2 better than Rubisco. The second advantage then is that this carbon dioxide, or this carbon cell, is eventually moved into the bundle sheath cell. And so the bundle sheath cell has sort of a different cell wall structure than our mesophyll cell, and this different cell wall structure makes it less permeable to carbon dioxide. So we can basically pack more carbon dioxide per volume gas in a bundle sheath cell than we can in a mesophyll cell. So in fact, we can store carbon dioxide in the bundle sheath cells in a way that we can't in a mesophyll cell. All of which is going to help our Calvin cycle, which occurs in the bundle sheath cell, run a little bit more efficiently. Okay, now what does this have to do with water loss? Well, if we have an enzyme that's better at grabbing CO2 concentrations at CO2 molecules at low concentrations, and then is better at storing carbon dioxide in these very specific cells, what it means is that plants can keep their stomata closed for more time during the course of the day. And so if plants can close their stomata for more hours a day, it means they have less water loss, which means they can do better. They can photosynthesize more in hotter and drier conditions. So this is a way for plants to sort of mitigate this trade-off between gaining CO2 and losing water. The majority of plants in our world have a C3 pathway, so it's worth noting maybe the exceptions and the plants that we are going to find in our C4 pathway and in our CAM pathway, which we'll look at in just a moment. In particular, there are a lot of grasses that are C4 plants. So corn, right, is a very common C4 plant, as is sugarcane. And here's a picture of sugarcane. So if you've never seen sugarcane growing, this too is a grass. It sort of looks like maybe a little bit like bamboo here with some very different looking leaves. Tastes a lot sweeter, unless you're a panda, I guess. We also can see, though, some much smaller C4 grasses, and crabgrass is a common C4 grass that we see all over the place in this part of the country, particularly in our lawns, especially in the mid to late summer. Crabgrass does very well if you've ever noticed the kinds of grasses that are growing in lawns around campus or maybe the lawn of your yard. Our third pathway is the CAM pathway. CAM stands for crassulacean acid metabolism. Don't worry, I'm not super interested in you remembering what these letters stand for, but I'll just point out that the Crassulaceae is actually the name of a plant family, and it's the family in which this pathway was first discovered. So we now know that there are several other plant families that have CAM pathways, but we retain the name CAM for crassulacean acid metabolism. So again, I've drawn a little diagram of what's going on in our CAM pathway and parked it next to our diagram of what's going on in the C3 pathway so that we can see the differences. So again, what enzyme binds carbon dioxide, or at least is the enzyme that first binds carbon dioxide? So once again, like our C3 plant, carbon dioxide is going to enter the mesophyll cell. So here we have carbon dioxide entering the mesophyll cell, but CAM plants, like our C4 plants, are going to grab this carbon dioxide molecule with PEP carboxylase. So that's the enzyme that's grabbing it. Remember that PEP is a 3-carbon molecule. We're going to combine it with carbon dioxide to give us a 4-carbon molecule, and we're going to create a variety of what are known as organic acids. So these are stable molecules that are going to hold on to this extra carbon that we've grabbed and stuck with PEP. The Calvin cycle, like in our C3 pathway, is going to be occurring in the mesophyll cell. So this is sort of a reversion from what we saw in the C4 pathway, right? So our Calvin cycle is still occurring in the mesophyll cell. No bundle sheath cells here. However, what CAM is doing that is somewhat different than the C3 pathway and the C4 pathway is that they are breaking apart the reactions of photosynthesis temporally. So the light reactions, of course, have to take place during the day in the light. The Calvin cycle also has to take place during the light because our light reactions are feeding it energy molecules, ATP and NADPH. But the capture of the carbon itself, right, so that the Calvin cycle sort of plus any other reactions that go with it, the capture of the carbon itself does not have to take place during the day. And in CAM plants, in fact, it takes place during the night. So I've tried to show that over here by putting this part of our CAM pathway, right, the initial capture of the carbon and its conversion into a series of organic acids. This is in our little block over here denoting the night. So these organic acids can accumulate all night. Then during the day, what happens is that these extra carbon molecules from the organic acids are released to the Calvin cycle. the carbon is grabbed by rubisco. We again get our 2, 3 carbon molecules, and away we go through the Calvin cycle. What are the advantages of separating the carbon capture in time from the rest of the photosynthetic reactions, the light reactions, and sort of that subsequent part of the Calvin cycle? Again, I'll quickly move over so we can see our advantages. The first advantage, of course, for a CAM pathway is the same advantage that we have in a C4 pathway, right? PEP carboxylase has this higher affinity for carbon dioxide than does rubisco. But the second advantage is, as you sort of alluded to, carbon is captured at night instead of during the day. And so what this means is that stomata can remain closed throughout basically the entire day. Stomata are shut tight, and yet the plant is busily photosynthesizing, even though CO2 is not coming in through the stomata at this point in time. Instead, of course, carbon dioxide is coming into the plant through the stomata at night, when it tends to be a lot cooler, when it also tends to be a lot more humid, particularly in tropical parts of the world, and so you get a lot less evaporative loss of water from your leaf into the environment. To show you what a CAM plant looks like, I've brought in my pet orchid over here. So, very proud of my orchid. I got it at a sale at Kroger, and it's been a wonderful orchid for several years now. When we think about CAM plants and plants that are trying to modulate this trade-off between carbon dioxide gain and water loss, we tend to think about plants that live in warmer environments, but maybe drier environments. And that's actually not the case. So while C4 plants can do well in drier conditions, many CAM plants actually live in the tropics, where it can be really wet. And this is the case for our orchid over here. So if a plant is living in the tropics, particularly a tropical rainforest, where there's all kinds of water, why does it need a CAM pathway? And the answer is that a lot of these CAM plants, while they're living around water, can't get a lot of it. And that's because, in the case of this orchid over here, it's an epiphyte. So an epiphyte is a plant that grows on another plant. So epi upon phyte plant. And so because it's growing on, so many orchids are epiphytes growing on other plants, they can't send roots down into the soil to pick up all this water that's sitting there in the soil. Instead, they produce these aerial roots, like this particular root right here. And I'll bring it a little closer maybe so you can see. No, the camera's not going to focus maybe. Well, so you can see, I hope, that the root is sort of fuzzy, and it's got this little velamen structure that's all over the root, and that sort of little hairy, waxy structure allows it to absorb droplets of water from the air around it, and that's actually the source of water for our orchid. So not a lot of it, which means that our orchid has to really do a lot to try to hang on to water. So if you've ever felt an orchid leaf, it's not sort of a papery leaf like we might have for a maple or beech or something like that, tree around here. It's sort of really thick, and that's to hang on to, to start hanging on to a lot of water. But of course, the other thing that it does to hang on to a lot of water is it's got this CAM pathway in which it only opens the stomata at night, not during the day. So what this means is that CAM plants tend not to be highly productive plants, in that they're not grabbing a lot of carbon dioxide per unit time. They're not adding a lot of biomass per unit time. But they can grow in some really remarkable places, and they can keep growing for a while. So our little orchid over here last produced a leaf. This is its most recent leaf over here. It last produced a leaf over the summer. So you can see it's not a big grower, but it is starting to grow, and periodically it also produces, of course, new shoots with flowers. So I'm very proud of my orchid. It's got a new little shoot. I don't know if you can see it right down there. All right, so it is in fact growing, particularly here at night, as you can see. Right now is a great time for it to take in lots of carbon dioxide. In addition to orchids, there are probably several other CAM plants that you have heard about. So if you've ever had a succulent plant, and as I watch people growing plants in their houses, including students in their dorm rooms, I see a lot of succulent plants. They're very commonly sold in stores because they don't need a whole lot of care. They can take a whole lot of abuse in terms of not being watered frequently, growing in kind of not well-lit places, but they've got these very thick leaves, And just like the orchid, they're very good at hanging on to water, and they're very good at photosynthesizing in lower light environments because they can take carbon dioxide in during night and then use light during the day, whatever light energy they have, to take carbon that has been stored from the previous evening. All right, so this is a typical little crassulacean plant. And here it's not growing in anyone's house. Here it's actually growing in a very challenging, hot, rocky environment with not a lot of great soil. Some cacti are also cam plants, but not all. Orchids, as I mentioned, are another great example of cam plants. Finally, a group of plants that you might see and even have eaten are pineapples. And so pineapples are bromeliads. Bromeliads are a family, so notice that for a pineapple plant, there's the fruit. It's a multiple fruit, remember? And we see these very thick leaves over here, indicative of cam plants. This particular bromeliad, pineapples, actually grow in soil, but like so many of these other cam plants, there are a lot of bromeliads that don't. There are a lot of bromeliads that are epiphytes. The tropics are full of epiphytic bromeliads. You just sort of look into the trees, and the trees are literally dripping epiphytic bromeliads. But in the United States, we don't see a lot of bromeliad diversity. One of our main bromeliads in the south is what we call Spanish moss. And so it's not moss at all. It's this great flowering plant. It's got a cam photosynthetic pathway. It grows on trees over here, such as oak trees, and then it dangles its stems and its very sort of thick, rounded, grayish leaves off the stem, and it does just fine. It can grow, as you can see, as you know maybe, for a long time on branches and very prolifically. Both CAM and C4 pathways are pathways that have evolved in response to trying to mitigate this trade-off between CO2 gain and water loss. And both of them do it in some very different ways. Both of them do it very successfully. But for every solution, there's still a trade-off. So even though we're mitigating one trade-off, we're creating another. And so I'd like to end our thoughts on photosynthetic pathways by thinking about the trade-offs that exist between C4 and CAM on the one hand and our more familiar pathway, the C3 pathway. Well, C4 and CAM plants do very well at capturing CO2 and hanging on to water. This comes at an energy cost. And so both C4 and CAM plants are more energetically expensive than C3 plants. In other words, it takes more energy in a variety of ways to photosynthesize. So let's now look at all three of our pathways together to see where this energy expense is being incurred. For C4 plants, they are running another cycle, right? So this is the cycle that first captures carbon dioxide using PEP carboxylase. And to run any cycle biochemically, you need an energy source, and that energy source, of course, is ATP. So some of the ATP that is being produced in the light reactions over here is now going to be needed to run this other cycle. So there's an energy cost, not to mention the cost of producing additional enzymes that make this cycle possible, and producing enzymes is also energetically expensive. So that's a cost associated with being a C4 plant that you don't have in a C3 plant. CAM plants, again, have these same sorts of costs. Again, to run another cycle over here requires ATP, so that's energetically expensive. And of course, it's also expensive to produce these acids over here that hold the carbon dioxide, and to produce the enzymes that grab the carbon dioxide. Let's look then, let's end by thinking about how these trade-offs between our pathways, C4 and CAM on one hand versus C3 on the other, might be impacted at different scales. Let's see how these trade-offs play out. Let's start with trade-offs at a small scale. And so this is a trade-off and an example that we can see right here around us, midsummer into the late summer, even into the fall. So we are known as the bluegrass state here in Kentucky. We've got yards, and we've got nice horse pastures full of lots of bluegrass. And if you pay any attention at all to grasses, you notice that bluegrass tends to do very well in the spring. But bluegrass doesn't do so well, particularly if you don't water it later on in the fall. And the reason is that bluegrass is a C3 plant. On the other hand, if the summers get and the late falls get really dry, then other grasses start to appear amongst our bluegrasses and can outcompete them, in particular crabgrass. So crabgrass grows much more than bluegrass during this later summer and fall period. And the reason that it does that is it's a C4 plant. So as the weather gets hot and dry, it does a much better job of capturing carbon dioxide from the atmosphere and hanging on to it, thus being able to close its stomata and hang on to water than our bluegrass does, right? So it can look green and continue to grow even when all the bluegrass around it dies in this late summer period. On the other hand, why don't we see crabgrass taking over yards and taking over fields earlier in the spring when it's cooler and we have lots of water? Well, that's where this trade-off comes in. And of course, it takes more energy to produce, to go through a C4 pathway than it does to go through a C3 pathway. So if we keep our environment cool, if we keep our environment very well watered, crabgrass then will be outcompeted, or at least sort of held to a standstill, by a bluegrass, which doesn't, or other C3 grasses, which don't have to incur these extra energy costs associated with a C4 pathway. Bluegrass, crabgrass, C3, C4 trade-off is one that we can see playing out around us on a small scale, but these sorts of trade-offs are going to also play out on much bigger scales, including global scales. So let me introduce you to what's going on in our world, in case you didn't know, in terms of what's happening with our CO2 concentrations and what's happening to our temperature. And then we'll think about how that's going to impact this trade-off between C3 and C4 pathways. So carbon dioxide concentrations, as you probably know, are rising. So in the next couple of slides, I'm going to illustrate how significant these carbon dioxide changes have been really in the last couple 100,000 years. And we can tell what carbon dioxide concentrations are doing in the atmosphere through ice cores. So if we drill into glaciers, as we drill down into glaciers, we go back in time. The most recent snow and ice was deposited, the top snow and ice was deposited most recently. The oldest ice is the ice at the bottom. So much like any sort of sediment core, we can get an ice core in glaciers. And ice, of course, as you know, also traps little air bubbles. So as we get older ice, we can also get older air bubbles and actually measure the concentration of carbon dioxide in those older air bubbles. So we can go back, we're getting pretty close to like 200,000 years ago in time, actually being able to measure CO2 concentrations in the atmosphere. So this particular graph shows CO2 concentration in the atmosphere as measured in different ice cores. And so this is time, and we're looking at CO2 over here. So notice that over the last 160,000 years or so, CO2 concentrations have fluctuated between a low of about 200 parts per million to a high of just coming up on 300 parts per million. All right, so certainly CO2 concentrations in the atmosphere do fluctuate. But what's happened more recently? We take a look not at the last 160,000 years, but the last 1,000 years, we see a very disturbing pattern. If we look back 1,000 years ago, and for most of this last 1,000-year history, our carbon dioxide concentrations have been relatively stable at about 280 parts per million. Then, during the starting with the Industrial Revolution and the larger scale burning of fossil fuels, coals, oils, etc., our carbon dioxide concentrations in our atmosphere have shot up. So this graph just goes to the year 2000, and so by about the year 2000, we're a little over 350 parts per million. So that's already a very significant increase in carbon dioxide concentrations of about 70 parts per million, or about a quarter of, we're increasing by about 25% our CO2 concentrations just from prior to the Industrial Revolution. This next CO2 curve shows what carbon dioxide concentrations have been doing post-2000, and so these have been directly measured at different observatories, such as the Mauna Loa Observatory on top of a volcano in Hawaii. So you get observatories that are very high in the atmosphere and can sort of very accurately record changes in CO2 concentrations, and so Notice that our CO2 concentrations are continuing to increase exponentially, past 380 parts per million, past 400 parts per million, and eventually, currently, our CO2 concentrations are just a little over 410 parts per million. So even in the course of my lifetime, we've had an increase in carbon dioxide concentrations of 60 to 70 parts per million, which is really astounding, staggering. If CO2 concentrations keep increasing at this rate, then over the course of your lifetime, we're going to be into the mid to upper 400s. So this will come close to being, by the end of your life, could even be a doubling of CO2 from what we had pre-industrial period. One final thing that's interesting about this second curve, however, is that notice that it fluctuates on a yearly basis. And so the reason that CO2 concentrations are fluctuating on a yearly basis at this observatory, which is in the northern hemisphere, is that the vegetation in the northern hemisphere is changing, right? During the summer, we have lots of plants growing. They're going to take carbon dioxide out of the atmosphere. During winter, most of these plants in the northern hemisphere are dying or becoming less photosynthetically active, and so CO2 builds up again. So this would be a winter, spring into summer, winter, spring into summer, and so on. So notice that plants have this extraordinary ability to take CO2 out of the atmosphere several parts per million just over the course of a particular growing season. Along with our changes in the atmosphere, we've also had significant increases in temperature concentration, again, in the past century and a half or so. So this particular curve shows deviations from a baseline. So this 0.0 represents our average temperature at, I think, 1900 over here. So we're deviations from the 1900 temperature. And notice that we've increased our temperature by about 1 degree C over the course of the past century. And in particular, the last 40 years or so has seen a very dramatic increase in temperature, ending with the last 5 years on record ending with the 5 most recent years for which we have full years of data. And these 5 most recent years over here, as you can see, have been the 5 hottest years that we have had on record. One more note about temperature is that changes in temperature around the surface of the Earth are not occurring equally in all places. So some parts of the Earth, including some parts of the United States, some areas over the ocean, are not warming nearly as rapidly as other places on Earth, particularly the polar regions, such as the northern polar regions, and the land masses around it, such as Asia over here, Eurasia in fact, is warming fairly rapidly. Parts of our own northern hemisphere, such as Alaska and Canada, are warming very rapidly too. And so while our average temperature increase in the last 120 years or so has been about 1 degree Celsius, in other parts of the world this temperature increase is coming up on 5 to 6 degrees Celsius, which is really frightening. I mean, this is ecosystem level changes in temperature that we're seeing. And so now we get to the point where we're thinking about how our behavior as humans, by increasing CO2 concentrations and temperature, is going to impact what plants do, right? At ecological levels, developmental levels, evolutionary levels, and so on. And this is going to be an important question, not just for the plants themselves, but also because we rely on particular plants for our own well-being. Particularly, we rely on plants to provide the vast majority of calories consumed by humans here on Earth. So, this is sort of one of the key questions that we're dealing with, that we're thinking about as a species as we think about this trade-off between different photosynthetic pathways, C3 and C4 pathways. So, will these increases in carbon dioxide concentration and temperature have the biggest negative effect on C3 plants, C4 plants, or CAM plants? And there are some interesting arguments to think that these pathways are going to be differently impacted by each of these different changes. So, for example, if we think about just carbon dioxide per se, putting more carbon dioxide into the atmosphere, as we're doing, might actually benefit C3 plants, because C3 plants have a little bit more trouble balancing this loss of water, they have to keep their stomata closed a bit longer. If there's more carbon dioxide in the atmosphere, then maybe they can get away with closing their stomata for a little bit longer and not paying this energetic cost that C4 and CAM plants pay to try to hang on to water in more challenging circumstances. On the other hand, global temperatures are increasing, and so as global temperatures increase, this is going to play to the strengths of C4 and CAM pathways. Because these plants, of course, are able to shut down their stomata for longer periods of the day or even all day to hang on to water and yet still be able to have the carbon dioxide they need to keep photosynthesizing. How is this going to play out then in terms of crop plants? Well, one of the major crop plants we have in North America is corn. So are increases in temperature going to be harmful or beneficial to corn? We'll see. Another plant, though, that provides a lot of calories for humans on Earth are rice plants. And rice plants are C3 grasses. Are rice plants going to benefit by higher CO2 concentrations, or will they be more negatively impacted by changes in, or by increases in temperature? And what helps rice mitigate increases in temperature is that rice paddies are often very moist places. In particular, rice paddies are flooded. So often rice plants don't have to worry about large losses in water. They're in standing water. But if our climate gets warmer and rice paddies are grown increasingly in non-flooded or less flooded areas, is this trade-off going to become a much more significant trade-off? And the thinking is that it might be. It's going to have a significant impact on our ability to feed large portions of the human population. And so now there's actually a growing research effort to try to genetically engineer rice so that it has a pathway more like a C4 pathway than a C3 pathway. And regardless of what pathways plants use, as temperatures increase, they might have to close their stomata more, and no matter what plant you are, if you're closing your stomata more, you're photosynthesizing less. So I hope this is kind of a very interesting way to end our discussion about plant behavior, particularly plant physiology, by thinking not just about plant physiology as it impacts plants, extraordinary as that is, but also thinking about how plant physiology impacts us, how it impacts our behavior and how our behavior impacts plants. So I hope you've enjoyed these lectures. I will have one more lecture for you on the fungi. They're a really neat group of organisms too, and then we're going to start thinking about the animals. And the animal lectures are going to be provided by Dr. Stevens.